Welcome to our lecture on receptor tyrosine kinases, RTKs. We will explore their structure, activation, and downstream signaling pathways. Additionally, we will discuss their involvement in development, disease, and potential therapeutic strategies. Let's dive into this fascinating field of molecular biology. In this section, we will explore the overview of receptor tyrosine kinases, RTKs, and their importance in cell signaling. RTKs are proteins that have enzymatic activity and are vital for transmitting signals inside the cell. They are involved in important biological processes such as cell growth, survival, and differentiation. Studying their structure, activation mechanisms, and signaling pathways is crucial in understanding cellular processes. Moving forward, let's delve into the structure and activation mechanisms of receptor tyrosine kinases, RTKs. RTKs are composed of an extracellular ligand-binding domain, a single transmembrane helix, and an intracellular domain harboring the tyrosine kinase catalytic activity. Ligand binding to the extracellular domain initiates a series of events leading to receptor activation. This activation involves conformational changes, receptor dimerization, autophosphorylation at specific tyrosine residues within the intracellular domain, and subsequent recruitment and activation of downstream signaling effectors. The activated receptor tyrosine kinases, RTKs, serve as platforms for various effector proteins. These effectors transmit signals to downstream pathways such as Ras-MAPK, and JAK-STAT, which regulate various cellular processes including gene expression, cell cycle, survival, proliferation, and differentiation. Moving on, let's unravel the fascinating concepts of crosstalk and integration of receptor tyrosine kinase, RTK, signaling with other pathways. RTKs often engage in intricate crosstalk with other signaling pathways, such as G-protein coupled receptors, GPCRs, cytokine receptors, and integrins, to fine-tune cellular responses. This crosstalk and integration of signaling events enable cells to integrate multiple extracellular signals and respond appropriately to ensure homeostasis and regulate complex cellular processes. Now, let's explore the intricate interactions between ligands and RTKs. Ligand-RTK interactions play a crucial role in determining the specificity and selectivity of RTK signaling. RTKs exhibit diverse ligand binding specificities, ranging from growth factors such as epidermal growth factor, EGF, platelet-derived growth factor, PDGF, fibroblast growth factors, FGFs, and vascular endothelial growth factors, VEGFs, to hormones and cytokines. The binding of different ligands to RTKs triggers distinct signaling cascades, highlighting the complexity and versatility of RTK signaling. Furthermore, let's dive into the concept of receptor dimerization and activation. RTK activation often involves receptor dimerization, either through ligand-induced homodimerization or heterodimerization with other RTK family members. Dimerization brings the intracellular kinase domains of two receptor monomers into close proximity, allowing trans-autophosphorylation of tyrosine residues within the activation loops. This autophosphorylation event positively regulates the tyrosine kinase activity and creates binding sites for downstream signaling effectors. Next, let's explore the intriguing mechanisms that regulate receptor tyrosine kinase, RTK, activity and the feedback mechanisms involved. To maintain cellular homeostasis, RTK signaling is finely regulated through several mechanisms, including ligand availability, receptor endocytosis, ubiquitination and degradation, and phosphatase-mediated dephosphorylation. Additionally, cells employ feedback loops to modulate the intensity and duration of RTK signaling, ensuring proper cellular responses and preventing aberrant signaling. Shifting our focus, let's investigate the profound roles of receptor tyrosine kinases, RTKs, in development, differentiation, and tissue regeneration. RTKs play critical roles in embryonic development, where they regulate cell fate determination, tissue patterning, and organogenesis. Furthermore, RTK signaling is pivotal for maintaining tissue homeostasis and orchestrating tissue repair and regeneration processes, such as wound healing and tissue regeneration after injury. Now, let's explore the involvement of receptor tyrosine kinases, RTKs, in cancer. Dysregulation of RTK signaling pathways has been strongly linked to tumor genesis and cancer progression. Genetic alterations, such as mutations, amplifications, and translocations, can lead to aberrant activation of RTKs, promoting uncontrolled cell growth, survival, angiogenesis, invasion, and metastasis. Targeting RTKs and their downstream effectors has emerged as a promising therapeutic strategy in combating various types of cancer. Moving forward, let's discuss the pharmacological interventions targeting receptor tyrosine kinases, RTKs. Small molecule inhibitors and monoclonal antibodies have been developed to specifically target and inhibit RTKs implicated in various diseases, including cancer. These targeted therapies offer personalized treatment options by blocking the aberrant signaling pathways associated with specific RTKs, thereby inhibiting tumor growth and improving patient outcomes. Shifting gears, let's delve into the intricate role of receptor tyrosine kinases, RTKs, in complex diseases beyond cancer. Emerging research suggests the involvement of RTKs in various complex diseases, including neurological disorders, autoimmune diseases, cardiovascular diseases, and metabolic disorders. Understanding the specific roles of RTKs in these diseases presents exciting opportunities for therapeutic interventions and improving patient care. Finally, let's explore the emerging trends and future directions in receptor tyrosine kinase, RTK, research. Advances in genomics, proteomics, and high-throughput screening technologies have significantly contributed to our understanding of RTKs and their signaling networks. Future research aims to unravel the intricate details of RTK signaling dynamics, identify novel therapeutic targets, develop innovative therapies, and enhance our understanding of intricate cellular signaling networks. Receptor tyrosine kinases, RTKs, are important for cell signaling, impacting many biological processes. Studying their structure, activation, regulation, and interactions with other pathways adds complexity and potential for personalized medicine and targeted therapies in complex diseases.